Medical Proteomics. We have 4 speakers listed over here, and our first one is Professor René Zahedi. He just got a professorship from the University of Manitoba. It's in Canada. Winnipeg. René, please come and start your presentation. This is all what I need to do?
Yeah, best don't do
The best don't do anything.
Okay. Yeah, thank you, Christoph, for inviting me. It's a pleasure to be here and present some data. Actually, most of the data, or actually all the data I'm going to present has been in Christoph's lab when I was still working for him. I just moved to Winnipeg 2 months ago, so I don't have any fresh data from there. The topic is immuno-multiple reaction monitoring, or as we call it, immuno-MRM, for the targeted absolute quantitation of cancer proteins in tumor tissue samples. So as you probably all know, in precision oncology, what happens if a patient presents at the clinic with a tumor? Usually these tumors are screened for genomic biomarkers, and if you detect one of these biomarkers, they are used to predict a specific treatment, a targeted treatment, for instance, with monoclonal antibodies that specifically target a protein, and hopefully abolish this regulated signaling. These targeted treatments have led to spectacular successes in the treatment of especially advanced cancers. But there are 2 major shortcomings that still need to be addressed. Recent publications show that only around 14% of the cancer patients in the US are actually eligible to these kind of treatments. So for most tumors, there are either no known drivers, or there are no drugs to target those drivers. And from those who are eligible for targeted treatment, only around 50% seem to be responding. So we have biomarkers that tell you that you are eligible for a treatment, but they don't necessarily do a good job. And those targeted treatments are very expensive. It's around $100,000 per patient per year. And altogether, this is a big market. Precision oncology is around $50 billion in 2019 and expected to triple by 2030. So there's more and more drugs, and they're getting more and more expensive, more and more patients eligible. So there's definitely something that needs to be addressed. As many people say, one of the reasons is that we're looking for genomic biomarkers, but there is definitely a disconnect between genotype and phenotype. As we hear in the proteomics session, I guess we are all aware of it, that proteins are the main drivers in cancer or in many diseases, but are also the main drug targets. So just looking at a genotype won't necessarily lead you to the right decision in treatment. And we expect, it has been shown actually that if you add transcriptomics data, you can actually increase the response rates of drugs, but it's expected that if you go to the protein level, measuring actually drug targets or also phosphorylations, you're going to have a better response as well. Another thing is that there are, of course, not only genomic biomarkers, also what has been used extensively in the clinic is immunohistochemistry. This is an example of anti-PD-L1 immunohistochemistry. I'm going to get back to that later. But immunohistochemistry has also some problems. It's not really standardized. It's not really quantitative. This is different sections from the neighboring sections from the same tumor stained with different kits for this particular target, PD-L1. And they're giving you different responses, different stainings, and therefore also lead to different clinical decisions. So this is definitely something that needs to be addressed as well. So we were particularly interested in quantifying specific low abundance proteins, in particular this pathway, the PI3-AKT-mTOR pathway, which is a central pathway in many tumors. And this is just an example of some data that we took from a paper from Matthias Mann from a couple of years ago for HeLa as a cancer cell line. It's not necessarily super representative, but still it's interesting that there are these super high abundance proteins that are in the millions of copies, but if you go into those proteins that seem to be interesting in the context of cancer, many of them are much lower in abundance and therefore probably difficult to analyze. So if you want to have an analysis of these kind of proteins, we definitely are interested in using mass spectrometry assays. And the things that we need to address, the requirements that we have to meet is we want to have assays that are standardized. That means if you're using them at different laboratories around the world, you would ideally want to have at least a similar, ideally the same result. You want to have an unambiguous identification, which is not necessarily the case for antibody-based methods like immunohistochemistry. You get a staining, but how can you really be sure that this is a specific staining and you don't have interference? In mass spectrometry, we can have interference, but based on the methods that we are using, we actually can see that there is interference. We want to have absolute quantitation, so we want to measure concentrations that makes it really able to analyze individual samples. So we don't want to have these classical proteomics workflows where you have controls in tissue sample, disease samples, and then you look relative quantitation and can classify them. We want to be able to take the sample from an individual patient and just from the data from that patient make a decision. And of course, we also want to be able to look at multiple targets at the same time instead of just looking at a single biomarker. This usually should lead to more interesting or more comprehensive results. And the way how we're going to do that is using triple quadruple mass spectrometers with microflow LC. And this is more robust than using like the newest state-of-the-art instrumentation, which is also more expensive. It's also more, these triple quadruples are also certified and used in a lot of clinical laboratories already, and the whole procedure is more cost effective than using like the newest instruments that, of course, are also giving you great results. An example I want to show you here is the tumor suppressor PTEN. It's the main negative regulator of the PI3-AKT-mTOR signaling pathway. Its main role is it dephosphorylates PIP3 back to PIP2 and therefore actually deactivates AKT, the AKT kinase. So PTEN is deregulated in a lot of different cancers and mostly downregulated in a lot of cancers. And it has been shown in breast cancer and colorectal cancer that this downregulation seems to be not connected to genomic mutations. And that's very interesting about PTEN is that it has also been shown that the degree of its downregulation is proportional to the severity of the cancer. So it's kind of a dose-dependent effect. So therefore, it's quite interesting to really measure it very accurately and precise. There has been conflicting data in the literature on whether it is a prognostic predictive biomarker for breast cancer, and it has been discussed in this literature as well that one of the reasons is that the methods that have been used to quantify PTEN are usually immunohistochemistry or Western blot, and as already mentioned, these methods are not necessarily standardized and they don't really give you reliable results in many cases. So this is definitely a good way to go for mass spectrometry, to have a more standardized assay. And our hypothesis was very simple, that if we're able to have a sensitive assay that allows us to quantify this protein very precisely in a robust manner, that we could be able to assess whether it is a prognostic or predictive biomarker. And the way we wanted to do it, as I mentioned already, using typical bottom-up proteomics approach, digesting, analyzing the peptides by triple quadruple mass spectrometry, so developing specific MRM assays that allow us then to quantify the protein using internal standards, so stable isotope labeled peptides that have the same amino acid sequence as the target peptide but are different in mass. And again, we want to use this with a high flow rate to make it more robust. So the way we did this, we selected a peptide that was unique for PTEN, that seemed to have a good response. We did a lot of different tests to make sure it also has a good digestion efficiency and so on and so on. And then we made a calibration curve to check, first of all, what's the limit of detection, limit of quantitation. We did this based on the guidelines from the Clinical Proteomic Tumor Analysis Consortium, or CPTEC. And we got, with our setup, to a limit of quantitation of 900 attomole on column. We have now a newer MRM system, the latest HLN 6495C, and there their sensitivity is even better with these high flow rates, but we have a very linear response, all good. We also did a repeatability test to check how reproducible this whole thing is if you repeat it 3 times per day on 5 different days. And we had an overall assay CV of around 15%. So this looks all good. We had a robust assay that had quite some nice sensitivity. But when we applied it to analyze tumor tissues, we realized it's not sensitive enough. We basically couldn't see the protein still. So that's why we then continued to set up an assay that we call immuno-MRM using antibodies, and this has been published last year. And the idea is that you, after digestion, you are still looking for the same peptide, but now you raise antibodies that are specific against this peptide. So you do an anti-peptide enrichment. And the beauty about it is that you can spike in your stable isotope labeled peptide to your digested sample and then use your antibody to enrich for both the endogenous and the standard peptide to the same efficiency, and then basically run your MRM assay again. So we using this together with a method that we also published recently where we use what we call 2-pick. So we use basically 2 different stable isotope labeled variants of the same peptide. So they all have the same sequence, but they have a different in mass in their mass shift. And we spike them in different amounts. And what happens is then you get a curve for your endogenous peptide, so the target that we actually want to quantify in the tumor. You have a curve then in blue for the one of your stable isotope labeled standards, cis1, and then one for the cis2, the other one. And that allows us then to do a simple internal calibration curve, and we tested this extensively, and actually we didn't see, it actually performed the same way as the classical external calibration, but has a lot of advantages, which I don't want to point out because of time restraints. One of them is actually that it's faster as well. So the actual assay that we use now, so we're having tissue lysate, we do tryptic digestion, release the peptide of interest, we spike in our internal standards at different concentrations, we then do the anti-peptide enrichment, and then do the triple quadrupole, and this allows us then to analyze it. So we tested this. First of all, what is the recovery of the immunoprecipitation? We did some optimizations and then tested it by spiking in different amounts of the target peptide into a background of colorectal cancer FFPE lysate, digest actually. And we could see that we have a good recovery of around 90% on average, so that's good enough, much better than. Actually, I have to say also, these anti-peptide antibodies are much better than anti-protein enrichment. For one reason, they give you much more specific signals, so we have less background, and you can also use more stringent washing and so on. So it's much more efficient than an IP on the protein level. And of course, we cannot control the IP on the protein level the same way we do it here. Then we also checked the assay accuracy. We spiked in recombinant PTEN protein into a sample that was basically devoid of PTEN. Again, a tissue sample. And then we checked what's based on what we know we spiked in, what amount we quantify. That looked pretty well. So we have around 87% on average accuracy, which is good enough. So we now know we have high recovery and high accuracy. And then finally, we use this assay to see how it performs. We measured first cell lines, different tumor cell lines. We measured a lot of them. These are just 3 examples. And so we have one that is actually, according to the Western blot, absent, so it has no PTEN. And this is a 50 microgram of lysate has been loaded here on this Western blot. And then we have 2 different cell lines with a little lower and a little higher PTEN levels. And then we measured the same cell lines using our assay. And what we can see here, first of all, the results kind of correlate. But what's interesting actually is that although we used only 20 microgram here as a total protein input, so less than on the Western blot, we can actually still quantify PTEN clearly in the sample that was supposed to be PTEN negative. So what we can see is that our method is already much more sensitive than the Western blot. Then we did the same with fresh frozen tissues. We used some colorectal cancer samples here. Only 3 patients, but we had also surrounding liver healthy tissue. These were liver metastases of colorectal cancer, and we had surrounding liver tissue, so healthy controls. It's not the perfect control because it's not the same tissue, let's say. But what we see here is that in the metastatic tissue, the PTEN levels were always lower, and you can see they're really low. It's 0.4 femtomole per 10 microgram of total protein input, so it's pretty low amounts. And in the healthy surrounding tissue, it was much higher. So now we were wondering, is that because the healthy tissue is liver and the tumor tissue is kind of colon? And so we checked actually the Proteomics DB database from Bernard Christoph, who's also one of the speakers here today, of course, because they have a lot of information on protein levels across different tissues. And we could see that the levels of PTEN in liver and colon were almost the same. So what we see here is definitely what we expect, that the PTEN is downregulated in a tumor control compared to a healthy tissue. And what's really interesting here is that the levels of PTEN that we see in those tumor tissues are certainly, the concentrations are definitely lower than the concentration in this specific cell line that was already negative in the Western blot. So there's another advantage of using our method, that we can see differences in samples that you don't detect anymore in Western blot. So next thing, we used this assay to analyze breast cancer PDX model, and we extracted fresh frozen tissue as well as FFPE tissue cores, and then compared our assay with Western blot immunohistochemistry. And this is shown here. This is now the concentration of PTEN in femtomole per 10 microgram using our assay, and then we have them sorted based on the Western blot results, and it kind of correlates. You can also see from triplicates here that we have pretty low CVs. That's also very nice. So we have kind of a correlation between Western blot and our assay, but of course our assay is more valuable because it gives you actual concentration instead of just some kind of arbitrary staining. Then we checked it with FFPE tissue, which is a bit more challenging, of course, and here we compared it to immunohistochemistry, which is basically shown here in the color code. Basically, there are samples that were deemed PTEN negative by immunohistochemistry, low PTEN, moderate or high PTEN based on the color code. And again, we sorted them and show the concentrations on the y-axis, and again, we see kind of a correlation. What's interesting now, if you look into some of the samples, so these two are biological replicates, 7A and 7B, shown over here again. We see pretty good agreement of the concentrations of PTEN in those two replicates. That's nice. But we also have examples like over here, P2A and P1. These are not biological replicates. These are two different tumors, or two different mice with different patients, let's say, sorry. And they are both PTEN negative according to the immunohistochemistry. But we can see with our assay, actually, they do both express PTEN, and there is actually an almost tenfold difference in the concentration of those two. So we are also more sensitive than immunohistochemistry, and so we think we can provide better, more information than the existing assays. So next thing, we checked whether how our data compares to the genotyping. So we looked at the gene copy numbers of the different tumors here. And there are two examples. We picked it here, patient number 3 and patient number 7, here in gray and in black, which have both the same gene copy numbers, but a total difference in the expression levels, both when we look into the fresh frozen tissue, but also when we look into the FFPE tissue, and also in the Western blot and the immunohistochemistry. So basically, again, it's just another confirmation that there is no correlation between the genotype and the phenotype in this particular case, at least. So we were interested when we used this for predicting response to drugs. We made a small study. Unfortunately, we don't have too much data yet, but this is an example where we used a PDX model of triple-negative breast cancer. We had primary and metastatic tumors, and they were treated with chemotherapy paclitaxel. And we then measured the best average response over 21 days, and according to the RECIST criteria that are listed here, so how well actually does the tumor shrink or not shrink based on the treatment. And then we did use FFPE cores to measure the concentration with our assay, and then we also looked into immunohistochemistry. And what we can see, there seems to be some kind of separation between the samples that are resistant to the treatment and in red and those that are responding. You see there is not a clear distinction. There's definitely an overlap. And this should not be saying strong, but we see a kind of a correlation between PTEN levels and response, but the levels are really, the numbers of samples are really too low yet. But these are for the metastatic tumors. We can see that if there is a higher concentration of PTEN, actually, the tumor seems to respond better to the treatment and shrink, while those that are very low in PTEN don't seem to respond. And then you can see the numbers are low, and it's not super robust, but it's a nice indication, and we're working on measuring more samples to get that. So to conclude this part, we developed a robust assay to quantify PTEN. We get a good agreement between the semi-quantitative results from immunohistochemistry in Western blot in our assay. We can distinguish even samples that are supposed to be PTEN negative by those two methods, and there seems to be no correlation between copy numbers, gene copy numbers, and protein levels. We see some correlation between the treatment response and the levels of PTEN, which we need to follow up now. And so we are trying to do this right now to get more statistical power. Now, in the next 5 minutes, I wanted to give you another example of this assay that we're using for another interesting topic in cancer. Is the PD-L1 axis, as we call it. It's very important for all kinds of solid tumors, particularly for lung cancer. We have around 25,000 new diagnoses in Canada alone every year. And out of them, 80% are tested for this protein PD-L1 with immunohistochemistry. And already, as I mentioned already, the treatment costs are very expensive. And the problem, so why is it interesting to look for PD-L1? It's actually part of the immune checkpoints that protect healthy tissues from an immune response. So what happens is your cells usually express PD-L1 and PD-L2, which are ligands for the PD-1 receptor on T cells. And when they interact with each other, these cells are protected from the T cell response. Now, the problem is that cancer cells hijack this mechanism and overexpress those two proteins, PD-L1 and PD-L2, and thereby try to escape the immune response. So you can block this interaction by using anti-PD-L1 or PD-1 antibodies. And these are very, very powerful treatments, treatments for cancer patients. So to be able to assess whether a patient is eligible for this kind of treatment, currently the state of the art is using immunohistochemistry. But as I already showed you before, the immunohistochemistry is not really standardized based on what kit you're using, and also clinicians don't necessarily agree on whether a patient is positive or not. There is a lot of discussions. So the main pathology expert in McGill said this is one of the biggest needs in oncology to have better assays for that. Also, immunohistochemistry is also affected by storage of tissues, so probably based on oxidation effects. So if you stain different adjacent sections of the same tumor that have been stored for different periods of time, you get a decrease in the staining. So we checked this with our assays, and we could see that we are actually we don't have this effect. So we can measure, we get the same results independent on how long these tissues have been stored. So so the thing is now that, as I said, it's not really a good biomarker, the immunohistochemistry. And what you can see here are different patients sorted based on their PD-L1 level, the tumor proportion score, as it's called. And those that are having high levels of PD-L1, from those only 50% respond to the treatment. By those that have basically zero almost PD-L1, 10% still respond. So we again have an assay that seems to classify patients based on how well they should respond, but it's not really predictive. So we set up an assay using, again, immuno-MRM to target in total 6 proteins of this axis, as we call them. I don't really have the time to go through all of this. Based on the T-cell response, we selected them. It's PD-L1, PD-L2, PD-1, and 3 other proteins that are basically part of the T-cell response. And this is the workflow we're using for formalin-fixed and fresh frozen tissues. We do deparaffinization, protein extraction. We added a deglycosylation step because some of these proteins are known to be glycosylated, and we're actually trying to assess to what extent they are glycosylated. We can do that with our assay. And then we do digestion using protein aggregation capture. So this is basically automated. We spike in our standards and then do a multiplexed enrichment. So we spike in 10 antibodies at the same time and then enrich those, and followed by mass spectrometry. So we did some extensive analytical validation of this method. I don't really have time to go through all of this, but we have a very good intra and interday repeatability, a good stability of the assay based on how it has been stored. We unfortunately have to go up to overnight digestion for this particular peptide. We hope we can do a fast digestion, but we see better recovery if we do longer digestions. And we get a very good whole assay reproducibility for the different target proteins. And then we used this to analyze a couple of tumor tissues. I think it was around 30 in this case, or maybe 39 or something like that. And we have FFPE and matched fresh frozen tissues. Unfortunately, I don't have the correlation plot here, but in the end, what you can see here is we get pretty consistent results between fresh frozen and formalin fixed, and we could also quantify most of the proteins in most of the samples. The most difficult one was PDL2, which is simply the lowest in abundance usually, and has also peptide that doesn't have a good response compared to the other ones. So what we did then is we compared the results from our PDL1 assay with the immunochemistry, and basically what we see is that there seems to be a moderate correlation between the immunohistochemistry and our assay, but there is a lot of samples that seem to be based on the immunohistochemistry PDL1 negative, but that clearly have PDL1 based on our assay. So we think that shows one of the limitations of the immunohistochemistry. And this is just an example where we have different tumors with their staining and the concentrations that we measured. And this example here is in discordance. You see less staining, but we can clearly see it has a higher concentration in our assay. So finally, before I come to the conclusions, what we are doing right now, so these samples were all unfortunately a bit old, so we didn't have clinical outcome data for them, so we're just collecting this data right now. But these were older samples that didn't have this treatment yet. So what we did with those, we could classify them based on our results into samples that were what we call immune high and immune low, based on the expression of the set of proteins. And based on this, we could actually separate them in their survival rate. So the immune high samples, based on our assay, had on average a better survival than immune low. So even without treatment, we can already kind of distinguish these kind of these kind of survival rates. What we're doing right now, I think we have around 200 samples, if I'm correct, that we need to analyze now, where we have clinical outcomes, and hopefully we will see that it will be able to predict the outcome. That's, of course, the goal. So to conclude this, we have an automatable, or it's basically automated, immunoassay for this PDL1 axis. In total, it takes around 448 hours, sorry, not 480. We see moderate correlation between immunohistochemistry and our assays, and we see that it seems to be good candidate biomarkers for the prognosis of non-small cell lung cancer. The clinical validation, as I said, is ongoing. And with this, I'd like to thank you for your attention. This is the acknowledgement slide. Again, I'd like to thank Christoph, who I was working with in Montreal for a couple of years, the people who did most of the work, Zahedi Ibrahim and Vincent Lacasse, and the funding agency. I also want to thank Ollie, who's in the audience, Oliver Pertz, who generated these antibodies. So if anyone is interested in anti-peptide antibodies, he's the person to talk to. Thank you for your attention.